Hello everyone, welcome back to our session on gene interaction. And in gene interaction, we have discussed about the allylic interactions. We discussed about three types of allylic interactions, which were complete dominance, incomplete dominance, and codominance, which was followed by the non-allelic interactions, where we saw two different genes and their alleles. They are interacting together to produce a common trait, and in that we discussed about complementary genes, which gave a phenotypic ratio of 9 is to 7 rather than the normal dihybrid phenotypic ratio of 9 is to 3 is to 3 is to 1. And in this session, we are going to discuss about another type of, or the second type of non-allelic interaction, and that is supplementary genes. So supplementary genes, this is also the interaction between two independent pair of genes, and they interact in such a way that, or their interaction is in a different pattern than that of the previously discussed complementary genes. And in supplementary genes, you can see that this is the interaction between two dominant alleles of two different genes. So in this, one dominant allele or the one dominant produces, that is a one dominant allele of a particular gene, it produces its effect in the presence or the absence of the other dominant gene. But the second dominant gene is so special in that it can produce its effect only in the presence of the first one. So first one is independent or one of the dominant gene. So here the interacting genes, the dominant alleles of this particular interacting genes, they are of the two dominant genes. One is independent and the other is dependent. The first one is independent in that it is having the capacity to produce its trait even in the absence of the other one. But the other one, or the second one, the second dominant gene of or the second dominant allele of the second gene is independent or dependent on the first one, in the sense that the second one produces its effect only in the presence of the first one. So such a type of interaction is called as supplementary genes. And when two dominant genes, or in this case when the two dominant genes come together, you can see that a new trait can be seen as an effect. So the main example of this is the coat color in mice. So what happens in this particular in supplementary genes is that when you're having two genes, for say I'm taking two genes as gene A, the dominant allele, and the other one as B. I take these two genes. So what happens in this case is that the one dominant, so these are two dominant, for say I'm taking it as both homozygous for this, and its recessive will be small b and small a. So I take now these two, the pair of dominant genes. In this case, what happens is that I take A as the first one and B as the second one. This A can produce, or A is having the capacity, or A is said to be independent in the sense that it can produce its effect in the absence of B. That means if B is absent, A produces its effect. A can produce its effect, or A can produce the trait which it determines in the absence of B. But B produces its effect, or when B to be expressed, it requires the assistance of A. Assistance in the sense that only in the presence of A, this B will be expressed. Only when capital A is present, capital B produces its effect. So we'll discuss about this, how they are interacting. So these are two different genes. So we are talking about a non-allelic interaction in the sense that these are two different genes and these are their alleles. The dominant one is A and the other one is small a. This is capital B and small b. So what happens here is, in these two different genes, they are going to, or they in non-allelic interaction, we see that these two different genes, they together will be contributing to a particular or a single trait. Or such a type of interaction is usually referred to as non-allelic interaction, and in that we are discussing now with supplementary genes. So now we are going to take the example of this particular example. In this particular example, we are going to talk about the coat color in mice, about the supplementary genes. So here you have two different alleles, as you see here, that is C gene as well as A gene, and you can see this one is dominant C, and here you are having a dominant A. So what happens here is we are going to talk about the interaction between this capital C and the capital A, and what will be their effect. So this is what happens here. You can see here when capital C is present alone. So this is a particular individual. You can see the two genes. Here A gene is recessive and C gene is dominant. And in this case, when C alone is present, you can see that C produces its effect. So this is the first gene that we talked about in the session of supplementary genes. This is the first gene. It is independent and it produces its effect. That is, it is producing the color which is called as the black color. But when you come to the other gene, the second gene, when this is present alone, alone is this case, only you can see capital A here. The other one is recessive. So when capital A is present alone, you can see that it does not produce its effect. It cannot produce its effect. It produces its effect, or it does not produce any coat color, and hence the coat will be, or it will be, the individual will be having a white coat color. And what happens here is when two individuals were crossed, one which was dominant, only homozygous dominant for one gene, the other was homozygous dominant for the other gene. So we know that one of the gene is independent; it expresses itself, but the other gene is dependent on C. It produces its effect only in the presence, only when C is present along with it. And that's why when A is alone present, it does not produce any effect, and hence the coat color will be white, which is called as albino. And when they are crossed, both capital A and capital C comes together in a particular individual, and that results in the expression of A. Okay, A will be expressed in that case, and it produces a new color or a new phenotype, and that phenotype is called as a gouty or a brown color. This brown like color is called as a gouty. So here it is said that this particular agouti color is dominant over black and albino. This agouti color, this is dominant. The coat color is dominant over black and albino. And if you take the dominant hierarchy, or the dominant hierarchy in this case, it can be seen that agouti, I mark it as A. Agouti is dominant, or it is higher than, it is dominant over B, and it is also dominant over the other one, which is A. So this is the dominant of this, and it is dominant to B, and it is also dominant to albino. So if you take the hierarchy, it will be like this. So this is for, I'm marking it for albino or white. So this is for albino. So albino is the one which is recessive to both black as well as agouti. Agouti is dominant over black as well as albino or white. So this is their dominance hierarchy. So this one is agouti is dominant over both black as well as agouti, and black is dominant to white or albino. So albino is the one which is recessive in this group. So these both are considered to be wild type one, and the agouti is found to be dominant over black. Dominant in the sense that most of the individuals will be having this agouti coat color, and that's why this is considered to be dominant over black. And what was seen is that when this particular F1 heterozygote was crossed, a new type of dihybrid ratio was obtained. As you see here, you can see here that in all the cases where A and C comes together, you can expect to have this brown or agouti coat color. When C alone is present, we said that it is independent, it produces black color. When only A is present, you can see that when only A is present and when both are absent, both the dominant genes are absent, it produces a coat color which is called as which is so this is the where the dominant genes are absent. So in all these three cases, you can see the only one gene is present, and that is A. In all that cases, you can expect the color A will not express its particular feature, and hence it will have the coat color which is called as white. And it gives you a new phenotypic ratio which is 9 is to 3 is to 4 rather than 9 is to 3 is to 3 is to 1. So here what we said is that here the dominant gene C is independent, and it produces a black coat color in the absence of A. The dominant gene A does not express in the absence of B, hence the coat color will be white. When both genes together is present, A as well as C, it produces a new coat color, and that is called as agouti. As a result, you can see that you will have a new ratio which is 9 is to 3 is to 4, and this is what is called as supplementary interaction or supplementary genes. So in supplementary genes, the main difference from complementary and supplementary genes that we discussed in non-allelic interaction is that the genes which are complement, the effect of each other and the presence or the genes which complement the effect of each other and the presence of both genes are essential for the production of the wild type phenotype. So we saw in the example that we took two white color plants, and when both the genes are present together, you express or you get a new color. In all other cases, what we expect is that when either of in both the case of complementary and supplementary, we are talking about the interaction of two different genes. In complementary interaction, the two genes, that means both the dominant genes are essential for the production of a particular trait. Neither of them is independent. When they are independently or when they are present alone in a particular plant or particular organism, what happens is that they do not produce a particular trait. They do not express. So if it is a flower color, if both the genes, both the dominant genes are present alone, they do not express any color, that will be white. But when both the genes are present together, they will produce a particular phenotype. That means both the genes are essential for the production of a particular phenotype. The absence of either of them will result in a trait which does not produce any expression, which usually if it is the flower color, it will be white. So both the genes are essential here to complement. That is why they are called as complementary. Both are required to produce a particular trait. But when you come to supplementary, which we discussed in this particular session, we said that one of the gene is independent but the other one is independent. So the two independent pairs of genes interacting in such a manner that one dominant factor produces its effect whether the other is present or not. We talked about the one which is called as the gene which is called as capital C, which produces the black color in the presence or in the absence of the other. So it is independent, it can produce the effect in the absence of the other. If the other gene is present, it helps the other one to produce another trait. So whether the other gene is present or absent, the one of the gene is independent, but the other produces its effect only in the presence of the other. So this one, in this particular interaction, to produce a particular phenotype, we said that there are three phenotypes, but here we said that there are only two phenotypes. Here the ratio was 9 is to 7, 9 colored one and 7 which was albino. And here we saw that the interaction produces a ratio which is 9 is to 3 is to 4. Three phenotypes were present. And in this three phenotypes, one phenotype was due to the interaction of the two genes. One was because of the independent gene and the other was because of the because of the second gene which did not produce any effect, which was albino. So here the dominant gene cannot produce independent traits. The dominant genes alone cannot produce. Here the dominant gene can produce independent effects which are different from the combined traits, that is black and the other one which is agouti. So here the phenotypic ratio is 9 is to 7. Here it is 9 is to 3 is to 7. So here the example is of the purple color flower. Here the coat color and also the grain color in maize is example for supplementary gene. So this is about the two type of non-allelic interaction. We are in the next session. We will be discussing about the third type of non-allelic interaction, which is epistasis. So we come to the end of this particular session. So we'll discuss about epistasis in the next session. Thanks for hearing.